Alright guys, let's get started. Alright, so we're continuing on the theme of transcription regulation. This is our second lecture. Alright. So why do we care about transcription regulation? Because it's one of the most important steps in which the genome is interpreted and basically deciding of whether to transcribe a functional product or not, right? So we said that the genome, if we depicted kind of a line like this, you're going to have your several genes here, and there will be several transcription factors binding to enhancers and promoters and regulating these genes. So in the last lecture, we talked about how you find where these transcription factors bind and what DNA sequences these transcription factors bind, because these sequences are the sites the transcription factors are going to search the genome, and when they find it, they're going to stably associate and regulate the genes, right? In this lecture, we're going to talk more about the transcription factors themselves, their typical structures, and how they may be also regulated. All right, the first thing that we're going to talk about is the fact that transcription factors are modular in structure. And what I mean by that is that for a given transcription factor, for a typical transcription factor, what you will find is that you will find a region of the protein, so a protein domain that can bind to the DNA, so we call this the DNA binding domain, and another domain which is going to be important for regulating transcription, and this could be an activation or a repressor domain depending on the transcription factor. So in the case of the yeast transcription factor GAL4, you have a DNA binding domain at the N terminus of the protein and the activation domain at the C terminus. The structure, the places that these DNA binding domains and activation domains within the protein can be different. So in this case, you have the N terminus containing an activation domain, and the C terminus has a DNA binding domain. And there are proteins with multiple activation domains as well. All right, so how do we know this, right? How do we know where the DNA binding domain of a protein is, or where the activation domain of a protein is, right? So we do know these through experiments that use reporters. So in this slide, we're going to learn about how can you find out where the DNA binding domain or an activation domain of your protein is using a reporter assay. In this example, we're going to analyze GAL4 transcription factor. So what you have here is that you clone your reporter gene. In this case, it's the LAC-Z, under a minimal promoter, which contains a tautobox, and the yeast enhancer, upstream activating sequences that are recognized and bound by GAL4. Okay? And now what you do is you also clone and express the GAL4 construct. What I mean by that is that you're going to produce a plasmid that is going to express a full-length GAL4, which is going from amino acid 1 to the end of it, right? And now you're going to have two assays. One is going to be an independent assay that's going to look whether your protein is binding to the upstream activating sequence or not. And these can be assays that we talked in the last lecture, right? It could be a gel shift assay, it could be a DNA footprinting assay, it could be any of these assays where you can say, does my protein bind to the DNA or not? And then you're going to put a plus when you see that it's binding and a minus when you don't see binding, okay? And then in the same assay, you're going to look at whether this protein is capable of binding and activating this reporter gene. So if you see the reporter gene being activated a lot, you're going to put a bunch of pluses. And if it's activated little, you're going to put just one plus. And if it's not activated at all, you're going to put a minus. Okay? So that's the assay that you're going to do. Any questions? All right. So next, what you're going to do is you're going to delete different parts of the protein, and now you're going to look at the consequences of that in the transcription activation of your reporter gene, asking, are these deleted regions required for the activation of transcription. So what you're doing is that you have your reporter, you're expressing all of these proteins. So if you express the full-length protein, what you're going to see is you analyze the DNA binding, it binds to the DNA, and it activates transcription, right? So it's going to be expressed, it's going to be binding to this upstream activating sequences and activate transcription. So when you look at this first deletion, in which you delete the first 50 amino acids of the protein and then analyze its DNA binding, you don't see any binding, right? And you don't see any activation. So does this mean that the amino acids 1 to 50 contain the activation domain? Can we conclude that? No, right? The reason why it's not activating is because it's incapable of binding to DNA, right? So if transcription factors lose their DNA binding domains, they can't go and find the sequences that they need to bind, and they're not going to activate it. Go ahead.
How do you know that you can cut off the activation domain?
You have to do multiple experiments, right? We can't say that this is the activation domain, and we can't also say, we can't definitively say that it doesn't contain any part of the activation domain. But the likelihood is that if you chop out the DNA binding domain, you're not going to see any binding, right? And the DNA binding domains and activation domains are separable, not the same part of the protein. They don't overlap, right?
Okay.
They're separable domains.
But if you cut off the activation domain, wouldn't you see the same effect?
Yeah, so now we're getting to that, right? So we know, okay, the first 50 amino acids are where the DNA binding domain is located, because when you cut it off, you don't see any binding. Where's the activation domain, right? So then what you do is you continue your analysis. You say, I'm going to make construct that contains the DNA binding domain, right? You have to have them, otherwise you're not going to see an activation. And then slowly delete more and more amino acids from the C-terminus, right? And so when you delete the amino acids between 848 and to the end of the protein, does it have any effect on activation? No, right? So I'm going to conclude that all of the amino acids between 848 to the end of it is not required for activation, therefore does not contain any sequences of the activation domain, right? And now you go further and further. What about between 823 and 792? Can be activation domain? Yeah, right? Because you're kind of doing something to the protein so that its activation ability is going down, right? And that's depicted by 2 pluses instead of 3, right? And then you go and delete a little bit more from 792 to 755. You reduce the activation activity even more, and then by the time you come to 692, you no longer have any activation, right? So from this, what can I conclude? I can conclude that the activation domain is somewhere between 692 and 823, right? From the experiments up to this point, that's what I can conclude, right? And then I'm going to say, let me just chop it up into even smaller pieces and then see what happens. So one of the things that you can do is you can see the minimal DNA fragment that is capable of binding to the DNA. So I'm going to make a construct and up to 74 amino acids and then show that it binds to DNA. It's good, and it's not going to activate because it's lacking that activation domain that I'm guessing is going to be right here, right? So then I can make further internal deletion mutants. So because these DNA binding domain and activation domain are separable, you can sometimes chop off all the regions in between them, and this transcription factor, the resulting protein, will still be a functional transcription factor, okay? So what you're doing here is you make an internal deletion between 74 and 684. And so what you would do is you would connect this with some kind of flexible linker. What I mean by that is that you're going to make a construct that is going to connect this, but it is going to be much shorter, and it's going to have 8 or 10 amino acids or so that will be flexible enough so that the DNA binding domain and the activation domain can still work. Okay? Does that make sense?
I'm still confused on how you can tell the difference between the DNA binding domain and the activation domain if when they're both deleted, function goes away.
You can't, and that's why we are having an independent assay to test the DNA binding activity of each construct at the same time, right? Okay, so you have a flexible linker containing and allowing you to make these internal deletions, right? So you're going to make more and more deletions, and then what you can see here is that from 684 to when you delete the amino acid sequences between 684 and 738, do you see an effect in activation? No, right? So the activation domain is not there, right? And then how about 738 to 768? Do you see an activation, an effect on activation? Yes, right? So you conclude that your activation domain is starting somewhere here, right? Somewhere here, 738, and also it's starting somewhere here, 823, because that's the first region that you saw an effect, right? And then so you say, okay, my activation domain is somewhere between 738 and 823 amino acids, right? Does that make sense? All right, so that's how you basically determine where your activation domain is going to be. Any questions? Good. We have added plenty of study questions on this, problem sets, that will help you practice this skill a little bit. It's kind of like a puzzle, right? All right, so these are the transcription factors, typically have two separable domains, and then we classify these transcription factors based on their DNA binding domains, families of DNA binding domains. Because these DNA binding domains have typical structures that can bind and recognize the sequences inside the DNA. And here are some examples of such structures. So this is the DNA binding domain from proteins that contain a basic helix loop helix structure. It's basically because of this, it has a helix, and then it has a loop, and then another helix, right? Like helix, loop, helix, and another short helix here. So these DNA binding domains typically dimerize, so that's why you see two of them. And then you have a helix that is binding to the major groove of the DNA, and this helix is going to make the necessary interactions with the bases of the DNA, and these interactions are going to be specific for each base, right? So it's going to make interactions with A or a T or a G or a C, etc. And then these interactions is going to determine what DNA sequence is this basic helix, loop helix is going to recognize. Here's another DNA binding domain that you see, which are zinc fingers. These are called zinc fingers because they need zinc ions to fold and bind to the DNA. And their structures are here again shown with a couple of beta sheets and a alpha helix. And again, they're sitting at the major groove of the DNA and then recognizing the DNA sequences. You have other DNA binding domains such as helix turn helix or a leucine zipper. So this is the DNA recognition helix, DNA binding helix again, and these are called leucine zippers because they homodimerize or heterodimerize through these interactions between the leucines. Okay, so when you look at an individual organism and look at the different transcription factors, you're going to have in humans, you have about 2,000 transcription factors, and each of these are going to belong to different families, okay? And this is one example in actually C. elegans, and we're looking at a zinc finger family of transcription factors here. And so they're listed here in kind of this relation tree, and these three are built based on how similar are the DNA sequences that are recognized by the two transcription factors. For example, in this case, CES1 and K02D7.2, right? And the sequences that they bind are very similar. So they have zinc fingers that recognize these two sequences. And if you actually line up the DNA sequence motif for this transcription factor against the other transcription factor and look at the amino acid sequences in the zinc fingers, you can kind of guess which amino acids are recognizing which of these DNA sequences and why this transcription factor is slightly different from this transcription factor is because that they have different amino acids in the zinc fingers recognizing different DNA sequences, okay? By just looking at the transcription factors and zinc finger domains, it's almost impossible to tell what the DNA sequence are going to be recognized by those zinc fingers. So there's a lot of experiments that goes into this kind of analysis. One of the things that you can do is you can crystallize the DNA binding domain with the a piece of DNA fragment containing the sequences that it binds to, and then see which of the amino acids are making these interactions, right? And then use that information to look at a related transcription factor and try to guess which amino acids will be important for the recognition of which DNA sequences, and then you can experimentally test your hypothesis or a model. All right, so what you have is that you have, in humans, you have about 2,000 transcription factors. A lot of them are actually zinc fingers, so more than 500 of these transcription factors are zinc fingers, and the other ones are going to be different classes like leucine zippers or homeodomain containing transcription factors, etc. So I just mentioned homeodomain containing transcription factors. These are, all of these DNA binding domains are extremely conserved. What I mean by that is that these DNA binding domains are the same type of binding domains that you would see from bacteria to humans, okay? And they work very similarly. What they do is they recognize specific DNA sequences. So the homeodomain containing proteins typically recognize a TA-rich sequence. So here we're looking at the crystal structure of the antennapedia homeodomain binding to the DNA. So the recognition helix is right here, again recognizing the sequences in this DNA. So homeodomain proteins contain a homeobox. A homeobox is basically a 180 base pairs long DNA sequence encoding for the homeodomains in transcription factors. And these homeodomain containing transcription factors are involved in the regulation of patterns of anatomical development, morphogenesis in animals and fungi and plants everywhere. Okay? And one of the most famous mutants that are typically used to look at mutations in homeodomain proteins is this guy here. It's the most famous fly in the world. So here's your normal fly looking pretty ugly, and then you have a mutant fly looking uglier because it has legs coming out of its head, right? And the reason for that is it has a mutation in the antennapedia gene, which is expressed in the wrong place in embryonic development. And now, because it's expressed and it's sufficient to drive the development of these legs, this animal has the unfortunate fate of having these legs coming out of his head, right? So these homeodomain proteins are also present in humans, and they do very similar functions. We have homeodomain transcription factors that regulate where our arms and legs come out, how many vertebrae that we have, how many fingers we're going to have, and all of that, these are determined by homeodomain transcription factors. All right. So while we're studying genome interpretation, we want to know which sequences. We learned about techniques such as linker mutation scanning assay that is going to tell us which sequences are important for the transcription of a gene, right? And if you know these sequences, sometimes you want to know which transcription factors actually bind to these sequences, right? as all of these deletion assays, linker scanning mutation assays, they're going to tell you this region of the genome is important for the activation of this gene. You're going to do all kinds of experiments and saying this sequence is important for the activation of that gene, but you don't know what transcription factor binds to that sequence. So you need an assay to do this, right? So one assay to determine which transcription factors bind to your sequence of interest is called yeast-1 hybrid assay. All right, so what are we doing in this assay? What we're doing essentially is we are preparing a reporter gene, reporter construct, in which your plasmid contains your reporter gene and the DNA sequence that you're interested in. Okay, so you're interested in what binds to this sequence here. So you clone this sequence upstream of a promoter and a reporter gene. And let's say that you suspect a certain transcription factor called protein X is binding to this sequence, and you want to test it. So what you do is you prepare a second construct in which you express the gene encoding protein X. And let's imagine that this protein X is a transcription factor. When it binds to your element of interest, it's going to activate the transcription of your reporter gene, and then if you see transcription, you're going to say, my protein X binds, right? And if you see no transcription, then you're going to say that my protein does not bind. So this is basically just a reporter assay to test if your protein binds to a sequence and activates the transcription of your reporter or not. Right? So this is the basis of the yeast-1 hybrid assay. But now let's talk about how the assay actually works. Let's imagine that the transcription factor that you want to test is in humans. Okay? So if it's in humans, then you express in the yeast with the reporter DNA element. Even if it binds, if it's not capable of working with the yeast transcription machinery, it's not going to activate the transcription. Does that make sense? So what you have to do is you have to trick this. And the way that you can trick this is, what you can do in the yeast is, instead of just expressing your transcription factor, you can fuse your transcription factor of interest to a yeast activation domain. Okay? So then your construct is going to be expressing this. It's going to express your transcription factor, and it's going to also have the fused activation domain. You can do this with the full-length transcription factor, or you can just take the DNA binding domain of your transcription factor and fuse it to a yeast activation domain. And it will work like a yeast transcription factor. Okay? So you can do this with one transcription factor if you suspect that this is one of your candidates, or you can use a library of transcription factors. What I mean by a library is you can build a human transcription factor library to be expressed in the yeast so that every human transcription factor DNA binding domain is fused to a yeast transcription factor activator domain. Okay? These libraries do exist, so you make that. So you take each of these plasmids and you transform it into the yeast that contain the reporter plasmid, right? And the reporter plasmid basically has your reporter here and then your sequence that you want to see which of these transcription factors are binding to, right? And we call this a bait sequence because we're going to use this bait to fish out the transcription factor that it binds. Does that make sense so far? All right. Next, what we do is we express these in the yeast. Okay. So in this case, our reporter gene is a gene. When it's expressed, it's going to let the yeast cells grow in media that is lacking histidine. Okay. So it's a Hist3 gene. That's the enzyme that is going to be used to make histidine. So these cells, if they are expressing the Hist3 genes, they're going to live on plates that don't contain histidine. So if your transcription factor fused to the yeast activation domain binds to this bait sequence, what it is going to do is the activation domain is going to activate your reporter. Is going to activate your reporter. So what you're going to have is you're going to have transcription of your reporter, and then these yeast cells can grow in minus His media. What happens if your transcription factor does not bind to the bait sequence? What is going to happen to those cells? If you plate them on, what is going to happen to history gene? It's not going to be expressed, right? It does not have the transcription factor binding that's upstream. It's not going to be expressed, and then these guys in minus his plates are going to be dead, right? Does that make sense? Question about yeast 1 hybrid. Okay. All right, so the next question we're going to talk about is how are transcription factor binding itself and function regulated, okay? Some of the themes that goes on here is the effect of chromatin structure on transcription factor binding, the fact that a lot of transcription factors bind combinatorially and regulate transcription combinatorially, and some of the transcription factors that can be regulated by ligands or modifications. All right, what's up with the nucleosomes? So eukaryotic genomes are wrapped around these histone proteins forming these nucleosomes, right? And this is important for DNA packaging and fitting inside the nucleus. But we also showed you some DNA binding domains of transcription factors that need to wrap around the DNA and bind to DNA and recognize these sequences in the major groove, for example. Is this structure compatible, for example, with the binding of this particular DNA binding domain? Does that look compatible? No, right? So generally, the nucleosomes are going to be inhibitory to the transcription factor binding in general. For those transcription factors that need to bind to DNA in a certain shape, and that is not compatible with this nucleosomal DNA, right? So, for example, leucine, I can't remember which family is this, but this DNA binding domain needs to, it's like a scissor, right, holding on to the DNA. That's not going to be able to bind to DNA if the DNA is wrapped around histones. So they are generally inhibitory. If they're inhibitory, then how do transcription factors find where they want to go, right? How do they actually get access to DNA? One of the things that we observe in eukaryotic genomes is that promoters in the genome contain usually less stable nucleosomes. So if you look at the nucleosome positioning across your genome, this is what you would typically find. You would find lots of nucleosomes covering lots of DNA sequences, but you will find that these DNA sequences at the promoters are typically more open. That doesn't mean that there is no nucleosome here. It just means that it's less stable. Okay? It's easier to fall off. So if it is less stable, then every time these nucleosomes are going to be falling off and assembled, the transcription factors can find a window of opportunity to bind. Give me one process in which every single nucleosome has to be removed from the genome. Replication, right? So it's going to create an opportunity for transcription factors to bind to DNA, even if normally this region is nucleosomal. There are also other transcription factors. There is a small class of transcription factors which we call them as pioneer factors. The reason why they're pioneer factors is they can bind to the DNA even if it's nucleosomal, and when they bind, they cause the removal of that nucleosome, removal of the histones, so that DNA is no longer wrapped, allowing for other transcription factors to come in, right? It's like pioneers going in, clearing everything, Historically, that's terrible, but in molecular biology, you clear everything and make room for the new transcription factors to come. And this is a very important mechanism that allows for activation of certain enhancers in certain cells and not the others, okay? So for example, if this gene is regulated by this enhancer, in most of the tissues, this enhancer is going to have closed chromatin. What I mean by that is that the nucleosomal DNA is not going to be accessible to transcription factors, but if in a certain tissue you have a pioneer factor such as TF2 binding to this nucleosomal DNA and removing that nucleosomes, allowing the binding of other transcription factors, these transcription factors can now start activating the transcription of this gene. And this is an important mechanism that involves, in development especially, for example, a very famous pioneer factor is called FOXA. It opens up the enhancers in cells to promote their development into liver cells. Okay? All right. So the transcription factors don't act just by themselves. For a given gene, many transcription factors control its activity. Okay, so what I mean by that is multiple transcription factors control one gene. So there's combinatorial control. So this figure is not depicting combinatorial control. It's just depicting a very simple situation. If it was that there was one transcription factor controlling one gene, to depress or activate that gene, you will create different cell types because you're going to have different numbers and amounts of the functional products. Okay, and so that's the basis of it. The combinatorial control is shown in this figure. So if there was only one transcription factor to regulate one set of genes, for 20,000 genes, you would need 20,000 transcription factors, right? Because if every gene had its own transcription factor. But that's not how it works. We have a small number of transcription factors that control multiple genes, okay? So if you have a combinatorial regulation, then you can increase the number of genes that can be regulated with a limited number of transcription factors. And this will also increase the range of regulation. So transcription regulation is not such that you turn on and off genes. It's kind of like a light dimmer. You tune up or down, okay? So don't think of it just on and off. It's usually up or down. And having multiple transcription factors is going to do this. All right, so how does this happen? For example, let's look at this example here. We have 6 sites. So let's imagine these are 6 promoter regions for 6 genes, okay? And each of these genes are going to use the transcription factors A, B, and C. Only 3 of them, right? And so the site A is going to be bound by transcription factor A as a homodimer, right? So when a transcription factor A is present in the cell, this gene is going to be turned on because it's going to bind, right? So that's the same idea for site 2. It binds to transcription factor B only. Site 3, it binds to transcription factor C only. So far, it's very simple. If you have A, you have site A bound, gene 1 on. If you have B, gene 2 on. If you have C, gene 3 on, okay? So let's imagine another gene now. It is controlled by transcription factors both A and B, right? So in cells that contain only A, this gene is going to be on, but this gene is going to be off, right? So even though this gene 4 is controlled by transcription factor A, so it contains the binding site for transcription factor A, because the transcription factor B is not present in cells that contain only A, this gene is going to be off or its transcription will be down, right? So same idea for site B, site 5. So again, it's controlled by A, but it also requires transcription factor C. So in cells that contain transcription factors A and B, this gene is going to be on, but this gene is not going to be on because you need factor C, right? So, well, if you add, this is just with a set of 3 transcription factors acting combinatorially with 2 only, you increase the range of transcription that you can achieve, right? All right. So you actually learned expression regulation of a prokaryotic gene by 2 transcription factors, so that the transcription is not just on and off, it actually can be regulated up and down. Can you guys remember what system you learned this? Combinatorial. You learned combinatorial regulation. Lac operon, right? The cap and the repressor, and together you could completely shut off the gene, express it a little bit, and then express it at a very high level, right? In response to different situations. Very good. All right. So what I mean by cooperation, okay? There is one case which is called close cooperation, where different transcription factors has to physically interact, and this regulates their binding. In other cases, the cooperation is indirect, such as the cap protein and the lac repressor. They independently regulate a gene, but the overall output is going to depend on the binding of both of these proteins. So here's a close cooperation example. So you have a DNA sequence that contains a weak NFAT binding site. So this is NFAT transcription factor, and a weak AP1 binding site. What do I mean by weak? What is it? A low consensus. So a weak site means these guys have DNA sequences that are kind of okay for the transcription factor to bind, but they're not optimal. They're not very similar to the consensus sequences that are bound by these transcription factors. So they're kind of weak. So their binding is going to be not very strong. So on their own, each of these transcription factors actually cannot bind each of these sequences. But together, what happens is that they make these physical interactions that change the DNA binding strength of their DNA binding domains, so that these 2 weak sites can be bound cooperatively. Okay? So now you have 2 weak sites that can only be bound in the presence of both transcription factors. So this can be in like this with physical interaction, or these interactions can be in a little bit more distant through the protein folding, so that you can have the 2 proteins binding to slightly different, slightly more distant sites, but again binding through these physical interactions. So this close cooperation examples are frequently seen in enhancers. So this is a typical, it's an enhancer example. So this is the beta interferon genes enhancer. There are the sequences that each of the transcription factor binds. So ATF2 binds to this TGAC sequence, IRF3, blah, blah. And each of these transcription factors binding regulates the activity of that enhancer. So what I mean by that, it regulates how much is that enhancer going to activate the transcription. Okay? So here's the model of how these transcription factors bind based on their crystal structure, and you can see in all of them can fit together to this enhancer. And some of them are not going to be affecting each other's binding directly. For example, P50 binding is independent of ATF2 binding, but together they are going to control how much transcription the target gene is going to have. Make sense so far? Okay, good. a very nice example of combinatorial transcription regulation is seen in neuroscience. So in neuroscience, there are many different types of neuronal cells, and each of these types you can tell because their structure and function are different, right? So if you look at this nerve cell, it doesn't look any similar to this one, and that one is very different from this one, etc., right? And each of these structures and functions are different, and all of these structural and functional differences are determined by the collection of genes that are transcribed from each of these cells. So the genes that are transcribed from this cell is different from that cell, etc., etc. And the combination of transcription factors determine which genes are going to be turned on. So here's one molecular example that will make it a little bit more concrete in your head. So in this example, what we're looking at is we're looking at how an LHX3 gene, the transcription factor, controls the transcription of genes that are going to be specifically expressed in the V2 type of interneurons versus motor neurons, okay? So the genes that are going to be expressed in the V2 interneurons, they're going to have DNA elements in which the LHX3 binding sites, the binding sequences, are going to be present and at a stereotypic distance from each other. In these V2 interneurons, LHX3 is expressed and another protein called NLI is expressed. And they form this heterodimer and a homodimer here, and that structure allows the recognition and binding to these type of elements, and they're going to now activate the expression of these genes containing these sequences. The genes that are going to be expressed in motor neurons are also regulated by LHX3, okay? So they're also regulated by the same transcription factor, but in this case, the location of these transcription factor binding sites, LHX3 binding sites, are going to be different from the V2 interneuron sites in that they're going to be more far apart. And they also require a third transcription factor called ISL1, which is going to complex with the NLI1 and LHX3 and bind and regulate the genes containing these type of regulatory sequences where you have both LHX3 and ISL1 binding sites. Okay? So you can see that although LHX3 is required for the expression of both V2 neurons and motor neurons, the way the molecular mechanism by which it regulates both of them are different and are determined by the presence of additional transcription factors and the location, the architecture, I would say, the location of where the binding elements are going to be in the regulatory sequences. Does that make sense? So you can imagine with 2,000 transcription factors and 20,000 genes, this is an amazing puzzle, right? So amazing puzzle is how each of transcription factors find their binding sites, where are their binding sites, and which combinations are regulating a certain set of genes, right? Okay, so that was basically, so far, all the things that I talked about in eukaryotic transcription factors is that if it's present, it's binding, right? But we know that that's not the case, especially for, we talked about prokaryotic transcription factors, the lactose or the cap protein, their binding is regulated by the presence of ligands, right? These ligands are going to bind to these proteins and change their DNA binding domain structure so that it can bind or cannot bind, right? In humans and in mammals, there are also examples of these ligand-regulated binding of transcription factors, and we're going to go talk about one of them, which are the hormone receptors. So in humans, hormones are very important regulators of gene expression, and there are different hormones here shown for cortisol, retinoic acid, etc. These are lipid soluble. They can go through the cell membrane into the cytoplasm. Okay, and their presence are recognized by these transcription factors called hormone receptors, and these different classes of hormone receptors. For example, estrogen receptor is going to bind to the estrogen, progesterone receptor, etc., glucocorticoid receptor, etc. And here are their domains. So in these protein transcription factors, you have a domain of these receptors that are going to bind to the ligand. So this is called the ligand binding domain. And they're going to have a separate DNA binding domain that's going to recognize a specific sequence at the regulatory regions of the genes. And then you have a variable region that is going to have variable functions. Okay? So what are the sequences these transcription factors bind? These are some of the examples of the sequences. What you can see is the glucocorticoid receptor. What you can recognize is actually the sequences. There are two sequences that are present, and these sequences are exactly the same, and they're separated by 3 nucleotides whose sequence doesn't matter. That's why they are labeled as N3, because you can have 3 nucleotides here. It has to be 3, but their sequence doesn't matter. And you have these sequences that are bound by the GREs, okay? The glucocorticoid receptor binds to DNA as a homodimer, and that's why you see these 2 sequences. So one DNA binding domain is going to bind to this sequence, and the other DNA binding domain is going to bind to this sequence, and the 2 glucocorticoid receptors going to homodimerize, then binding to regulatory regions that contain this sequence. Estrogen receptor is also a homodimer, and you can see that again, the sequence here and here is exactly the same. Again, they need to be separated by 3 nucleotides. These guys form heterodimers, and you can kind of see that. Maybe you can't from the sequence, but they form heterodimers, and they're going to bind to DNA sequences that look like this. But in this case, you can see that they have exactly the same recognition sequence, but their specificity is controlled by how many nucleotides can be in between these 2 DNA binding domains, these 2 DNA recognition sequences, okay? So the number of nucleotides in between is going to decide whether a vitamin D receptor is going to bind or a thyroid hormone receptor is going to bind.
What if the wrong dimer pair is set up? Like, does it throw one of them off? Or
yeah, so if it's not stably binding, it's going to fall off, and it's not going to be able to activate transcription.
okay.
Right? Any questions? All right. So these hormone receptors regulate transcription, and they do so by interacting with co-activators. So in eukaryotes, co-activators could be SAGA complex, right? It could be TF2D. It could be switch SNF. So that's how these activators are going to activate transcription. And the way that they do that is they physically interact with the co-activators. Okay, so here's one example. So here you see the estrogen receptor shown in this color, and it's binding to the estrogen here, shown in gray. And we call the estrogen an agonist because it's a substance that fully activates the receptor that it attaches. And that binding to estrogen actually allows this green alpha helix of the estrogen receptor to bind to the alpha helices from the coactivator shown in purple. Okay? So when bound to the estrogen, this conformation allows the binding to the coactivator. Now we look at the same estrogen receptor structure in the presence of an antagonist. which is a substance that attaches to the receptor but does not activate it or kicks out the agonist. So when the tamoxifen is actually bound to the estrogen receptor, what it does is it shifts the position of this alpha helix so that it covers the place where normally would interact with the coactivator. So this estrogen receptor will no longer interact with the coactivator. So in the presence of the tamoxifen, the estrogen receptor will not be able to activate transcription. Okay? So that's how an agonist and antagonist can regulate this. Now we're going to talk about a second level of regulation of these hormone receptors, which is their localization in the cell. Okay? So eukaryotes contain the cytoplasm and the nucleus, right? And the transcription factors, for them to work, they have to be inside the nucleus. If you have a transcription factor present in a cell, but it's only localized in the cytoplasm, would you call this transcription factor active in that cell? No, because it's not going to bind to DNA. If you don't have anything bind to DNA, you're not going to activate anything, right? So what the cells do is they have a mechanism in which an inhibitor protein binds to the glucocorticoid receptor at the ligand binding domain in the absence of the hormone. And this inhibitor binding keeps this hormone receptor in the cytoplasm. Okay? When the hormone is present and it goes inside the cell and binds to the ligand binding domain, the ligand binding domain structure changes in a way that it can no longer bind to the inhibitor protein, right? And when it's not binding to inhibitor protein, this glucocorticoid receptor is translocated inside the nucleus, and it's going to find the DNA sequences it binds to. And for hormone receptors, we call these things response elements. So it's going to bind the response elements in the regulatory regions of the genes that it's going to activate, and it's going to interact with the coactivators, then activating the expression of this gene. So how do we know this, right? How do we know that these proteins are regulated by translocation into the nucleus? We know this through a bunch of reporter assays. So I'm going to show you one example here. So what you want to know is, where is your transcription factor, right? So you need a reporter to tell you where the transcription factor is. So what you can do is you can fuse your protein, in this case glucocorticoid receptor, to your reporter gene. So that is reporter gene encoding for beta-galactosidase. And you have an assay to visualize where this reporter gene is, right? That's what reporter genes do. There are genes that are easy to visualize or have an assay for. And then what you can do is you can fuse your whole glucocorticoid receptor, or you can take parts of your glucocorticoid receptor and fuse it to your reporter construct here shown in purple. In this case, I'm only showing you one because that's the experiment that's going to work. That's why. So we're using the ligand binding domain and fusing it to the reporter assay. And then you're going to express each of these proteins in cells and check if they're nuclear or cytoplasmic upon your hormone response, right? So what you're doing is you're taking your cells, you're expressing your proteins. In this case, this is your negative control, in which you're only expressing your reporter gene. And this reporter gene, beta-galactosidase, its location should be indifferent to the hormone, right? Because we know that its localization doesn't change. It's always cytoplasmic. So in the absence of the dexamethasone, which is a glucocorticoid hormone, you expect your negative control to be localized to the cytoplasm, and that's essentially what you see. These little blobs here is the nucleus. And then when you add the hormone, you see no difference in the localization. It's still in the cytoplasm, right? If you fuse your reporter gene now to the full glucocorticoid receptor, in the absence of the hormone, the protein is cytoplasmic, right? You see all the signals cytoplasmic. But when you add the hormone, bam, you look at this, it's inside the nucleus, right? It's translocated. So this protein is localization inside the cell is regulated by this hormone. What about your experimental, in which you're trying to find out which protein domain is responsible for regulating that translocation, where you fused your ligand binding domain to your reporter, and in the absence of the hormone, you see that this reporter is in the cytoplasm, and in the presence of the hormone, what happens? It's in the nucleus, suggesting that this domain is sufficient, right, to make this reporter gene to be responsive to the hormone addition. So that ligand binding domain is required and sufficient. Not required. It's sufficient for translocation. Yeah.
Does the protein that brings it into the nucleus, is it on like the nuclear wall, or how does it work?
I don't know how it's, so you guys are going to learn in MCB2 a lot more, the cell biology aspect of these regulations. So there are, in the cells, there are proteins that regulate the translocation of other proteins through the nuclear pore. So it's usually, for most proteins, not all, going through the pore is a regulated process. It doesn't just walk in the pore, okay? For a lot of proteins, there are shuttling proteins that are going to keep it in. And there are proteins like the regulator protein that I showed. If they are bound to the protein, a specific protein, it prevents its translocation through that nuclear pore because it prevents those shuttling proteins interaction and binding to them, and it can't go in. Does that make sense?
Yeah.
But the specifics of what are the shuttle proteins and how they work, you guys are going to learn that later. Any questions? All right. Okay, so in the last 15 minutes, we're going to talk about how the enhancers and promoters work. This is actually a very active area of research right now because in the human genome, we first sequenced it, right? 2000s. We know the sequence. Fine, but we don't know how the sequence works, right? Then we say, okay, we need to map where the enhancers and promoters are. And in the past decade or so, we developed lots of assays, high throughput assays, and we can pretty much tell you where the regulatory genes, all of the regulatory sequences are in the genome. But that's still not enough, right? We can tell you where these regions, where these regulatory regions are. So for a given cell type, I can tell you where enhancers are. And we can tell these enhancers because they have distinct chromatin structures marked by specific histone modifications. So if we could map these specific histone modifications, then we can tell this is the region of the genome that's regulating something, right? That's the information we can get. But we can't tell you which gene is that sequence, is that enhancer is actually regulating. So it turns out there are thousands of enhancers, and then each gene can be regulated by multiple enhancers, and each enhancer can regulate multiple genes, right? So this is a complex puzzle in which we need to match each enhancer to each gene, etc., right? So we are not, so right now, the one of the challenges in the field is that how do you know the specificity of an enhancer-promoter interaction is achieved? So how do enhancers interact with promoters? Through looping, through the mediator complex, right? Because enhancers interact with promoters physically by looping, and that is mediated by the mediator, right? So easy to remember. So what other kind of proteins mediate these interactions? What proteins mediate DNA looping? SMCs, because they have that awesome circular structure that can hold two sites together, right? So we know that basically in eukaryotes, the way that enhancers and promoters work are through DNA looping of enhancers to the promoters, and these are mediated by a bunch of proteins that include mediator and the SMC complex, such as cohesin. These proteins, they can be actually other proteins as well, like coactors and coactivators and transcription factors can also mediate these looping, even in the absence of a cohesin, or even in the absence of a cohesin. So why do we care, right? Why do we care knowing about this? So I'm going to show you one example of a highlight. This is a publication from a few years ago now, where I saw, and I was like, wow, this is amazing. I wish I could have done this. I was so jealous. But this is an awesome work of a bunch of people. So what they were trying to look for is they were trying to look if enhancer-promoter interactions are behind some of these developmental diseases that we see, etc. So what they did is that they were looking at this a bunch of patients in which they have brachydactyly, and I believe that's basically the length of the fingers can be different, right? So this is one example where the length of this finger is actually shorter, and there are other examples in which other fingers can be short. So what they did is they isolated DNA from all of these patients and then looked for common changes, okay? So there's what could underlie this problem. And they found a region of the genome which contains this Wnt6, IHH gene, EphA4, and PAX3. And each of these genes, this is a large region of the genome, this is the telomere, this is the centromere, and each of these little gray boxes is a gene, okay? So this region contains all of these genes. And when they look at these patients, what they saw is that they happen to contain multiple deletions that removed the chromosome segment from here to here, okay? So the first hypothesis is that you could look at this and you can say EphA4 genes, lack of EphA4 is responsible for this disease, right? Because it's deleted. But the puzzle was it was not. So there were people with the deletion of this gene that did not show this phenotype, right? And you could delete this gene in mouse, you also did not see this phenotype. So the lack of this gene is not the problem. And the other way that they knew that is that there were patients in which the segment was actually flipped. So you had a chromosomal segment inverse. In that case, the EphA gene is not removed. It's expressed, but these people still had brachydactyly. Okay? So it's not that gene. So what's going on, right? So one thing that they had known was that from other experiments that these dashed lines here, they correspond to regions that contain insulators, right? We talked about insulators just a little bit in heterochromatin. What do insulators do in that case?
Maintain stability.
What is it?
Maintain stability.
They prevented heterochromatin spreading, etc., right? So insulators are the regions of the genome that are bound by these proteins, insulator proteins. And what they do is they separate the chromatin interactions and activity of two domains. So they insulate what's going on here in this domain from what's going on here in this domain. Okay? So they knew that these insulators were there. And what they recognized is that the deletions tended to delete this insulator right here. Okay? All right, what next? What next? It's hard to do experiments in humans, right? I mean, it's hard and it's probably not ethical, right? So they need a model, right? So what's amazing is that this region of the chromosome seems to be very conserved between humans, so the humans, all of these genes shown here, and the mouse, right? Mouse has a very similar structure. It has a whole bunch of genes at this region of the chromosome, right to the centromere, and it has an EphA gene with a very large non-coding region around it, PAX3 gene, and the telomere, right? So now what they can do is they can do I'm going to make the exact same deletions in the mouse genome that resembles the deletions in the humans. So they used CRISPR-Cas9, which we're going to talk about in the next few lectures, to make a deletion that mimics the deletion in the patient. So it removes this insulator here, and it removes the EPHA gene, right? And then they say, do we see the same phenotypes in the mouse? And mouse are just like humans, right? They have fingers, so we can look at them. And what you can see, which is amazing, is that in a wild-type mouse, so these are the different length of each of the mouse fingers here, and then what you can see is this deletion caused a brachydactyly in which that you have the one of the fingers is now short, right? Awesome. Now you have a model in which you can study this disease, right? So what do? They want to understand what's the molecular reason why you have this brachydactyly. So what they did is, one thing that they did is they looked at the expression of this PAX3 gene, okay? So they knew that PAX3 gene is involved in development of limbs. They said maybe the PAX3 deletion is causing misexpression of PAX3. So they used an assay that you already heard about, which is in situ hybridization, right? So they're looking at where is PAX3 expressed in the mouse. So in the normal wild-type animals, you see that the mouse limbs here developing usually doesn't have much PAX3 at all. But in a brachydactyly deletion, when you have this deletion, what you see is you see a very high expression of PAX3. So it is ectopic expression. Ectopic means in a place where it's not supposed to be. So incorrect expression of PAX3 in this mouse, right? So the next thing that they did is they mapped all the enhancers in this region, right? So they used a set of techniques. They said they found an enhancer that was upstream of EPHA4. And normally what this enhancer does is it activates the expression of EPHA4, okay? But when you have this deletion, which also deleted this insulator, right? So in wild type, this insulator prevents the connection between this enhancer and this promoter, PAX3 promoter, so it cannot loop over. But when you have this deletion, now this enhancer is capable of upregulating PAX3, so it's causing its induction, okay? So when that enhancer is upregulated, then you have the transcription of PAX3, and they concluded that the removal of that insulator caused misfolding of an enhancer to the promoter, therefore misregulating a developmentally important gene, right? So this is an example of how we should figure out which enhancers are talking to which promoters and regulating which genes in order to understand how the genome is interpreted to produce us, right? Okay, so I'll see you on Wednesday, and take a look at the study questions on lecture 18 and 19. We added a few more to give you an opportunity to solve some problem sets, okay?